I'm Aditya. I'm a grad student at the University of Basel in Switzerland, and I'm happy to present to you some work from my PhD thesis titled Variation in the Genetic Architecture Along Species Elevational Ranges. So species have restricted geographical and elevational ranges that often align with climate. However, they respond to climate warming mostly by range shifts and rarely by evolutionary niche expansion. Several theories have tried to explain why this might be the case, but one neglected and understudied aspect is the idea of non-independent inheritance and expression of traits that could lead to something known as genetic constraints. These genetic constraints are often summarized in the genetic covariance matrix, also known as G. And for example, here in populations distributed along an elevational gradient, you could have a situation where populations at the range edge have G matrices that are sort of aligned with selection, where selection is represented by beta here. And here in this situation, we see that when beta and G max are sort of concordant, we have a strong response to selection represented by R. However, if beta were orthogonal to G max, what we get is a small and weak response to selection, and evolution is said to be constrained. This paradigm brings me to my two main research questions today. The first one being, how does the genetic architecture vary along elevational gradients? And the second one being, to what extent is evolution constrained at species range margins? To answer my questions, I study 6 species of Brassicaceae that are distributed along elevational gradients in the Swiss Alps. 3 of them are alpine, the 6 species are spread across 3 genera. Within each genus, I have an alpine species and its lowland or subalpine congener. There are several advantages of working with Brassicaceae, and the first one is that they are competition averse and inhabit patchy areas usually to avoid competition. And they have, as a clade, lost associations with mycorrhiza, which means that their realized biotic niches are likely to be quite similar. All of the species that I work with are predominantly autogamously selfing, but one, Notsea corymbosa, and all of them are also conveniently diploid. Right. So to get into my sampling strategy in some more detail, this picture that you see here is a nice representation of my typical field site where I collected the plants, and you see in the background the beautiful Matterhorn. And so basically what we did was we hiked up this mountain and similar mountains, and we collected the lowest elevation population that we could find, and something from the highest along this transect, and something in the middle of this transect. So sort of to represent populations from the lowest species range margin in this mountain, something from the highest point as well. What we then did was we took these maternal seed families, so the maternally collected seeds, we raised them in the greenhouse for 1 generation to make up some full sip seed families. We also did this extra generation in the greenhouse to sort of minimize effects of site of origin. We took our full sip seed families and raised them in a heat and control treatment in the greenhouse. The heat treatment was characterized by a 30-degree peak around midday. We then, in summary, we raised about 29 to 35 maternal seed families for each population treatment and species combination, and this amounted to about 4,500 plants that we phenotyped in the end. We measured a few different traits. The first class of traits that we measured were growth traits. We tracked the growth of all the plants to estimate the growth rates, the asymptotic rosette size, which is the final size of the plant, and the time to fastest growth, or the time to reach the midpoint of growth, which sort of signals the maturation time of the plant. We also measured several 3 leaf functional traits, known as leaf area, specific leaf area. This sort of signals the amount of leaf built up per unit resources, such as carbon or whatever. And then leaf dry matter content, which is the ratio of dry and wet weight of a leaf. This is a good metric of the leaf construction costs and also tells you about how much water is contained in all of these leaves. So you might be wondering why these traits and why an additional thermal treatment. Previous macroevolutionary studies on 100 Brassicaceae species in the Swiss Alps have shown that shown a lot of trait divergence in these traits between high and low elevation species, and warming, so high and low elevation species show really divergent responses when they're grown under warmer conditions. In general as well, alpine plants are very, very sensitive to warming, so this sort of treatment made sense. And finally, the G matrices that I will be estimating based on these traits, I also wanted to sort of know how robust those estimates are to the environment in which they were estimated in. So yeah, so from all of these traits, I estimated separate G matrices for, or the genetic covariance matrices for each species, population, and treatment combination. And this sort of results in about, I think, 18 or so different matrices, yeah. So I'm left with the task now to compare the genetic architecture along elevation ranges. And this is not the most trivial thing to do, but, and I will be presenting some ways in which I did that. The first one is the effective number of traits, or the effective dimensionality. This is somewhat a direct measure of constraint that accounts for covariance structures among these traits. What this is basically is that you can decompose your data into the smallest number of n orthogonal dimensions wherein trait variation is distributed equally. So for example, in this nice spherical case, we see that trait variation is distributed equally among all three axes, and this results in an effective dimensionality of 3. This is also what is known as a spherical G matrix. But if you were to constrain variation along one axis, such as the x-axis, you kind of go down in the effective dimensions that you have. And in this case, you have this cigar or oblong G matrix with an effective dimensionality of 2. So on to my results. So the high elevation species are always on the top row, and their congeners are right below them, right under them. What's striking here is that there seems to be an equal amount of constraint no matter what species, population, or treatment it is. What I would like to draw your attention to is that given the fact that I measured G matrices based on 6 traits, if variation were distributed equally along all axes, then I would have an effective dimensionality of 6. However, what you see here is that systematically, no matter what species, treatment, population it is, you have a much lower effective dimensionality, somewhere around 3, 3.5. This sort of indicates that there is a similar level of constraint in all of these species, populations, and treatments. However, this makes no comments about the orientation of the correlation structures in space. The next thing that I estimated was evolvability under selection. I will preface this by saying that betas were simulated randomly. However, given the fact that they were uniformly distributed on a 6-dimensional hypersphere, or essentially this is numerically equivalent to expected or average evolvabilities, and sort of also signals the amount of total genetic variance in these, also signals the total genetic variance. What we see here is that for this species, maybe you can see my pointer, I'm not so sure, but Arabis cerulea and both Notsea species show similar evolvabilities along elevation gradients, but the highlighted species here, this is where we systematically see that the lower elevation populations have reduced evolvabilities and also reduced genetic variances. So now I've shown you that there are similar levels of constraint in terms of the dimensionalities, and that some species have lower elevation populations with reduced evolvabilities and reduced genetic variances. So I'm left with wondering, is it just reduced genetic variance that could contribute to reduced evolutionary potential in these populations, or is there some fundamental difference in the covariance structures in these populations? And to do this, I use something that is called a covariance tensor analysis. Essentially, what you can do is you can take all of your G matrices and you can bunch them up and make something called a covariance tensor, which is the higher order analog or a three-dimensional analog of what a G matrix is. This tensor contains like the covariances of covariances and sort of, essentially what you can do with this is that you can decompose this spectrally just as you would do any G matrix, and you can identify the axes of differences among G matrices. So how exactly do these G matrices differ is what I answer through this. So what these graphs show here now is the genetic variance along axes in which G matrices are supposed to differ. I will skip through the top row because they don't really have significantly different axes, and also Notsea brachypetala on the bottom right, I think. But what I will focus on are these two species that are highlighted here, where we see significant axes of variation among G matrices. And the higher elevation populations or the higher elevation populations usually had more variation in this direction, which led to differences among G. Why is that the case? So probing into this a bit further, for Arabis ciliata, what I see is that the divergent axis was highly correlated with the Gmax of the high elevation population, and that this axis was characterized by high contributions from SLA and growth rates. For Cardamene hirsuta, I could find that there were multiple axes that were correlated with Gmax and other principal components of the middle and high elevation populations, and that these were all together characterized by high contributions from leaf area, size, or growth, or some of these. So to conclude, my first question, which was how does the genetic architecture vary, I find some sort of a mixed answer. I find that for some species, it's fairly stable. For some others, there is variation. More interestingly, I find that in general, you might say that given that the effective dimensionality is equally low, maybe they're all equally constrained, but at least for some low elevation populations of the mentioned species here, there is even more reduced evolutionary potential. And for Cardamene residifolia, it seems to be only due to reduced genetic variances, but for the other two mentioned species, Arabis ciliata and Cardamene hirsuta, we see that it's not just reduced genetic variance, it's also fundamental differences in their correlation structures. And with that, I would like to acknowledge my research group, my supervisor, Yvonne Wheatley, and well, all of the people who helped me phenotype the plants, because this was a lot of work, and also the field assistants who helped collect all of these species. Thank you.
I was wondering for how long have these populations been in these environments, because I was expecting to see less genetic variation in their own environment as compared to the other environment, right? And you don't find that. You don't find an effect of the environment in the genetic variation of the different plants.
Yeah.
So I was wondering, what is your explanation for that?
So I think I also mentioned, so all of these estimations were done in the greenhouse. Initially, they were in a common garden, which mimicked the control conditions that I showed, but also then I raised these full subseed families in both a heat and a warmer condition along with the control. What I think that is happening here is that, in general, theory says that G matrices or covariance structures should be robust to the environment in which they are measured, but there are some papers and some theories that discuss that this might not be the case in some situations. What I gather from this is that my estimates of G are fairly robust to the environment, at least a G matrix that is composed of these traits, and yeah, that's about it. Maybe I would like to add that although I know for a fact that heat affects the means of these traits, what is interesting, therefore, is that the covariant structures are preserved no matter which environment they were raised in, and maybe that answers your question.
Hey, good morning, everyone. It is my pleasure to be here. My name is Yehu Moran, and I will be talking today about our work on how venom shapes actually the interspecific interaction of animals with their predators and also an evolutionary trade-off. So basically, venom can be any mixture of proteins which is injected from one species into the other. It typifies animals. It can be just a handful of toxins, or sometimes even in extreme cases, a few thousands of toxins. Venom is interesting from evolutionary and ecological point of view because it is, at the end of the day, shaped by the natural history of the species. It is very metabolically expensive, and it's a complex trait. So all those things together make venom an appealing model system for studying evolution. And we study it in sea anemones and not in scorpions, snakes, or wasps, or any other famous venomous animal. And the reason why we work on sea anemones, and specifically Nematostella bactensis, is because it's a very nice model organism. We can grow it in the lab, and we can genetically manipulate it, what essentially cannot be done on other venomous species. And basically, we spent more than a decade working on the Nematostella venom system. We showed that actually the venom changes dramatically between different life stages, and this is also supported by a large variety of different cell types that produce different venom components. And if we look further, basically what we see, sorry, one second. Okay. So basically what we see is that there is one major venom component in the adult sea anemone, which is called NV1. So it is more than 10 times more common than the next toxin in the adult. And you can see it in the upper right corner in the graph. That's a logarithmic scale. And NV1 is short peptides. It's 47 amino acids. It's cysteine rich. And it's a very potent sodium channel modulator, meaning that it's causing pain and paralysis in the stung animal. And I should also mention that unlike the initial expectations, it is not produced in stinging cells that typify all cnidarians, such as sea anemones, corals, and jellyfish, but it is actually made in epidermal gland cells that you can see in red in the lower right picture. So this is done with immunostaining, and you can see very clearly in red that those gland cells are really epidermal, and they're secreting NV1 into the medium. And sorry, one second. Yeah. So basically, we wanted to see now what's going on with NV1 in nature, in different populations, and what can really help us here is that the Nematostella has very well separated populations, actually starting east of here in Nova Scotia in the north, in Canada, going all the way down to Florida in the US, and there is very little gene flow between those populations for different ecological reasons I can mention later. And basically, what we saw is that NV1 is made by a lot of different gene copies sitting in one cluster in the genome. This is something we know for many years now from the lab strain. But when we looked on the different populations, we could see that actually this composition changes dramatically. So each box in this diagram is a single copy of NV1, and you can see that most populations have multiple copies in this cluster. This is making a lot of sense if you need to make a lot of NV1 simultaneously for secretion. But in Florida, it's very surprising that it's a diploid. We sequenced the whole genome. And in this specific specimen, we had a heterozygosity for the locus. And you can see that there is basically one copy without any NV1 and another copy of the chromosome just with a single copy. This is negligible. And when we look on proteomics, we can see in Florida versus North Carolina, which are actually quite genetically similar, we can see that Florida is making almost no NV1, whereas North Carolina is making tons of NV1. And this is actually the most differentially expressed protein in the whole animal. So this is proteomics for the whole animal, and you can see that NV1 is really an outlier. That's a 500-fold difference, which is a huge difference in proteomics. And now we wanted to understand better the ecology behind this difference. And actually, Nematostella is a predator, but it's also being predated by other animals. And these are the species that are found with Nematostella, the predator species found with Nematostella throughout its native range. So this is the grass shrimp, Palamenthes, and the mummy-chog fish, Fundulus. And this is not a gazelle-cheetah relationship, but here sometimes the gazelle eats the cheetah. It all depends basically on how big and hungry are the animals when they meet. So like each of them can eat, at least under lab conditions, can eat the other. And what we did, and this was done by a very talented postdoc in the lab, I should mention Joachim Sum. So he was doing it in collaboration with a group of Adam Reitzel at North Carolina University at Charlotte. And basically, this was a work done with Sidney Birch, who's a postdoc at Adam's lab. And what Joachim and Sidney did was actually looking on the interaction of the grass shrimp with Nematostella. And what you can see, this is comparison of the North Carolina animals with the Florida animals. The higher the number is on the y-axis, the more interaction you have between them. And what you can see is that the interaction is significantly higher for the Florida animals compared to the North Carolina animals. So basically, the shrimp interacts more, and this actually makes a lot of sense because the Florida animals don't have NV1, right? But this is just a correlation, and we wanted to test it experimentally. So what we wanted to actually see is whether NV1 plays a role in this difference in the interaction, and for different technical reasons, specifically for NV1, we couldn't use CRISPR. So Joachim came with a very clever idea about how to use synthetic microRNAs. I will not get into the technicalities. If you wish, I can explain you later how it was done over coffee, but basically he was able to knock down NV1 very efficiently with the microRNA expression. So both qPCR and RNA-seq showed a big difference, more than 10-fold, and at the proteomics level, we could show 8-fold difference. That's not 500, but 8-fold is still a lot in biology. And then, quite remarkably, we can see that the transgenic, well, the mutant line actually recapitulates the same difference compared to the control as the Florida and the North Carolina lines. You can see the graphs here, it's significant, but I think it's much more impressive to actually see the videos. So let's start from the one at the upper right corner. These are the control animals, and what you can see, I hope, is that the shrimp is really doing its best to avoid the sea anemone. So the sea anemone is burrowed in the sand, and the shrimp is really doing its best to swim above it and avoid it. And let's see how the transgenic animal looks, the mutant knockdown, and you can see that it's very, very different. So here, what happens is that basically the shrimp just stomps over the anemone. It basically really doesn't care much about the anemone, and 1 second, I'll try it again. So basically what it does, it just, it bites it a bit. Apparently it's not very tasty, but then it stomps over it. It's really not very much afraid of the anemone. So really a huge difference in the behavior, and this is, as you can see, this is actually mediated by NV1 levels. So this was actually making a lot of sense, but then we wanted to see what's going on with the fish, with the mummy-chong. And here we were really surprised because we saw exactly the opposite. So we saw both for Florida compared to North Carolina and for the mutant compared to the wild type, to the control line, we could basically see that here actually the interaction is reduced. So without NV1, what happens is the opposite of what happens with the shrimp. Now the fish is actually getting less attracted to the anemone when there is no NV1. This was very surprising for us, but it was a good lesson because we had to dig deep into the literature. And after digging into the literature, what we could find is actually a paper from 1988 from Ron Naib at the University of Georgia, showing that basically so Naib was looking into native populations in Georgia, and he could show that when there is a lot of fish, you get less shrimp, you get more sea anemones. So actually, the Nematostella gains a lot from attracting the fish because the fish is the predator of the predator. So by using the toxin for attracting the fish, the anemone is getting rid of its major predator, which is the grass shrimp, which is pretty cool. And well, it's a symposium about trade-offs, and I didn't show you any trade-off until now. So now it's the time for the trade-off. And we basically expected, since we know and we measured physiologically that NV1 production has a huge metabolic toll on the anemone, we expected that something would change the moment that NV1 is removed. And this is actually what we show here. So first, we measured it under normal lab conditions, and we could see no difference between the mutant and the control line. But then we realized that anemones in nature are eating far less than in the lab, so we moved them actually to starvation, what we call starvation, which is in fact a very minimal diet. And under the minimal diet, we could see that actually the animals which are mutant for NV1, not having NV1, are basically growing much better. So this was very noticeable after 2 weeks of this very minimal diet, and after 3 weeks, after 4 weeks, we couldn't notice a difference anymore, but this was because what happened is that the animals without NV1 actually split. They basically divided asexually into 2 anemones, each of them, and this happened in a huge difference compared to the control line. And also sexual reproduction, which happens in the Nematostella, was significantly elevated in the mutant animals. So without NV1, you actually get much better reproduction, both asexually and sexually, and this is clearly a trade-off. So that's an evolution trade-off between making venom and making progeny. So just to sum up, I showed you at the DNA, RNA, and protein level that there are differences between different Nematostella populations with venom composition. The major venom component NV1 is differentially expressed. And basically, we could show that this is actually manipulation of NV1 levels and natural variation in NV1 make a difference in their interaction with fish and shrimp, which are predators and predators of the predators. And basically, this really shows the trade-off between NV1 and venom composition and basically the ability to reproduce. And our future plans is to test it also for other toxins and other venom components, and also to understand better how the NV1 is used in order to attract the fish. This is a very interesting question for us. And with this, I would like to thank the organizers and thank you for listening, and of course, thank all the students and the collaborators who helped our project. And I'll be happy to take a couple of questions, or maybe one question if there's enough time. There's not enough time, so I'll be happy to get coffee with you and hear your questions later. Thank you very much.
Good morning, everyone. My name is Saubhik. I'm a final year PhD student at the Evolutionary Immunology Lab, working with Dr. Imroz Khan. And today I'm going to talk about immunity in the context of aging and evolution. So let me start off by saying that advanced age is associated with decline in most of the physiological function of an organism, and immunity is also not an exception. It has been shown that aging leads to adverse changes in immunity, and this is known as immunosenescence. Now, immunosenescence has a few distinct features. For example, you can see that invertebrate immune responses like the antimicrobial peptide response or PO response, they actually increase with age. Now, you may think that due to an enhanced immunity, organisms will be surviving better, but that is not the case. What happens is that infection leads to inflammation, and if inflammation persists for a long time, it can become chronic and can lead to death of the organism. This is known as immunopathology. So in this example, you can see that renal secretion through Malpighian tubule of beetles actually decrease after infection and decrease more with age. So this is because there is some damage to the Malpighian tubule itself by the overactive PO response. In a recent study in our lab, we have found that antimicrobial peptides, which are known to have a very specific role against specific gram type of bacteria, also lose their specificity with age. So in this experiment, what we did, we actually infected Drosophila melanogaster knockouts for antimicrobial peptides with gram-negative bacteria Providentia redkeri. Now, on y-axis, we have hazard ratio, which is a proportion of individuals which are dying after infection as compared to the controls. And you may expect that a higher hazard means higher death. So when flies, they did not have Drososin, you can see that their hazard is pretty less. That means there may be some other antimicrobial peptides which is acting in the fly to defend against Providentia redkeri. Same goes with another antimicrobial peptides, mechnicoin, but when flies did not have diptericin, you can see that their hazard is pretty high. And in another mutant which did not have any antimicrobial peptide but only diptericin, you see that their post-infection survival has been rescued. So we can say that diptericin acts very specifically against Providentia redkeri infection at a young age. However, at an old age, you can see that no matter which antimicrobial peptides the flies have or they don't have, all of them actually succumb to infection. And obviously, they all have a higher hazard. So all of these boils down to the fact that there is increased susceptibility to infection with age. And we can then say that immune function, which actually defends against ingression, pathogen ingression, is itself subjected to age-dependent deterioration, and an excessive immune function can lead to inflammation and disease-related death. So now the question is, if all these features of an aging immunity are maladaptive, why are not they purged by the force of natural selection, and why do they persist in population? And can the theory, evolutionary theory of aging explain such phenomena? So what exactly is evolutionary theory of aging? The evolutionary theory of aging suggests that before the onset of reproduction, the strength of natural selection is so high that it does not allow much harmful alleles to be in their population in greater abundance. However, after the onset of reproduction, the strength of natural selection steadily declines until it hits the last stage of reproduction. And theory suggests that organism starts to age from the onset of reproduction. Now, keeping this theory in mind, what Michael Rose and colleagues have done is that they hypothesize that if they could experimentally manipulate the timing of reproduction, then the rate at which the strength of natural selection will decline will also vary, and organism will evolve a differential fecundity mortality plateau. In simple terms, they will age at a different rate. So what did they do? They took a bunch of flies. With one group of flies, they allowed to reproduce and contribute to next generation just after their eclosion. And you can expect that the strength of natural selection will decline at a much faster rate. On the other hand, with another group of flies, they allowed them to reproduce and contribute to next generation 14 days after eclosion. So you can expect that the natural selection will decline at a slower rate. And after several generations, what they, after several generations of evolution, what they found that the early reproducing flies, they have actually evolved a faster aging rate and with a total lifespan of approximately 40 days, whereas the late reproducing flies actually age slower with a total lifespan of around 60 days. So now the question that we asked is that since aging affect immunity, do we see an earlier manifestation of immunosenescence in the faster aging ACOs as compared to COs? To do this, what we did, we infected flies from both these regimes, COs and ACOs, with gram-negative bacteria Providentia retigeri with a range of pathogen doses starting from 2.5 OD to 40 OD. We measured three things. One, first we measured that for how long they are surviving after infection. Second, what we measured how many bugs are there in the body of the fly. So the bacterial load will give us an estimate of their ability to resist, the host's ability to resist the bacteria. And third, we also tried to find some mechanistic insight behind the observed phenotype. So we did some transcriptomics assay. So now, as I told that COs and ACOs, they have a differing aging rate and they have a differing total lifespan. So now in order to have a comparison between these two, what we did, we measured their percentile lifespan, and we infected these flies starting from 1 percentile of lifespan to 80th percentile of lifespan. So 1 percentile of lifespan being the reproductive window of ACOs. You remember that ACOs actually reproduce early. 20th percentile is when COs reproduce, so we have the range, and also 80th percentile when both ACOs and COs, both of these flies are aged. So what did we find? Okay, so we actually infected both females and males, so I'll show you the female data first. Now, this is the same hazard ratio graph. On x-axis we have age and on y-axis we have hazard ratio. Just to reiterate that a higher hazard ratio means higher death. The red line here, the red dot and the red line actually denotes the early aging ACOs and the blue one denotes the late aging COs. So you can see that during their reproductive schedule, the early aging ACOs, they actually have a lesser hazard, okay? So that means they can survive infection at their reproductive window. But at a late age, you can see that at 80th percentile, their hazard actually goes very high. On the other hand, COs, they have a higher hazard, relatively higher hazard at when they are very young. But after that, their hazard actually doesn't change that much. So you remember we measured the bacterial load also. So what we did, next we plotted the hazard function, hazard ratio as a function of increasing bacterial load in order to measure immune tolerance. So you can expect that as the bacterial load will increase, hazard function will also increase, and a higher slope means a lesser tolerant genotype. So we measured that. So over here you can see that on y-axis we have increasing hazard and on x-axis we have increasing load. And these facets are for different age points. And this is a female data. So we can see that this increased hazard in the late aging flies is possibly due to their inability to tolerate the infection. However, after that, they could tolerate the infection in a like similar format. However, when we measured the tolerance of the early aging flies, now remember that early aging females actually showed a higher hazard at a late age. This is possibly due to their inability to tolerate the infection, not only that. So we found that they are better able to resist the infection at a later age and possibly pay a cost of immunopathology at a late age. Okay, so next we measured and plotted the infection response in the males. And very interestingly, we found out that males from both the regimes, they could actually survive infection at their respective reproductive window. So you can see here that the red line, again, so this is the tolerance graph, and you can see that for ACOs, which are the early aging lines, they have a very high tolerance ability at their reproductive window over here. And on the other hand, COs, which is the blue line over here, they have a high tolerance ability at their specific reproductive window. So we really got curious about the this male data, where we actually found out that their immunity is very different depending on their age. So in order to have a mechanistic insight behind this, we did some transcriptomics. Now, with the transcriptomics data, what we did some network analysis to find the differential, if the differential gene expression correlate with the observed phenotypic changes. So when we compared the males from both early and late aging regimes at 1 percentile, when both of them are pretty young, so we actually did not find any change in their immune gene expression. As you can see that the sham and infected, there is no difference. However, we did find the change across ACOs and COs, which this is because we found changes in male mating and sterility factors. Next, we measured ACOs at their 1 percentile and 80th percentile of life, and we actually found that at 1 percentile of life there is not much change between not much changes in immune genes between the sham and infected individuals. However, at 80th percentile, when they are pretty aged, we found that there are quite a few non-specific immune molecules which are getting expressed in these flies. Lastly, we measured the COs at 1 percentile and 20th percentile of their life, 20 percentile being the reproductive window of COs. And you can see that the change in immune gene expression pattern actually changes not only after infection but also with age. And this is majorly mediated by the immune regulators and metabolic genes at their 20th percentile of their life, suggesting that it is a controlled immune expression during that time. So to summarize, so since ACOs, they develop faster, we may expect that their immunity will mature faster, and by resisting the pathogen, they could survive infection at a very early age. However, the cost of which we can see at a later age in terms of higher immunopathology. On the other hand, COs, they actually, since they have a reproductive window after, so they have to maintain their health for a longer time, and it's possibly better for them not to allocate their energy in a very high energy intensive resistance mechanism, but into a tolerance mechanism. So I thank you for listening to me. I thank the organizing committee for inviting me. Thank the lab members for their inputs, and I'm open to questions. Also, I'm attending my pre-synopsis next month. I'm open for postdoc positions. Thank you.
Hello. Thank you for being here. I'm going to talk about a very famous trade-off that is called the RNK trade-off. But first, let me start by introducing the people that have contributed to this work. So this work started during the master thesis of André Mira, embarrassingly long ago. He was co-supervised by Leonor Rodrigues and myself, and we have been doing this work in collaboration with Klaus Rufler from the University of Uppsala and Inês Fragato from our own university. I also like this slide particularly because it shows the way that at our university we communicate with the rest of the world. I hope you appreciate it. So my career has been haunted by trade-offs, so I'd like to thank the organizers for telling me that it's still valid today to think about it, because I've been thinking about this for the last embarrassingly long number of years, and it has been still difficult for me to really come up with a solution. So the reason why I have been interested in trade-offs is because I cannot imagine biodiversity without the existence of trade-offs. So if there would not be a trade-off, we would have what we call a Darwinian demon, that is an organism that is able to adapt to all environments, which obviously would be a bacteria, which was the first organisms that appeared. And we know that the world is not like that, luckily. To me, I cannot find another explanation but the fact that it is not possible to be well adapted to all environments simultaneously. So there should be trade-offs somewhere, and I've been trying to look for them for a very long time in my life and never found them. So one of them that we decided to dedicate ourselves to is the RK trade-offs. So the RK trade-off states that So organisms that live in environments that are unsaturated have a particular set of traits that allow them to develop fast, and they are short-lived, they have small size, and this allows them to fast colonize faster unsaturated environments, in contrast to organisms that adapt or live in saturated environments, for which another set of traits that is contradictory to this one would be advantageous, such as big size, being good at competitive abilities, etc. So this has been originally proposed by McCarthy and Wilson in the famous biogeography book. So it was something between species. There is the corollary within species in many different flavors and many different theories, such as competition colonization trade-offs or the pace of life syndrome, etc. But in general, the idea is that there is this trade-off. So people have tried to test whether this trade-off exists. They have done so. I'm talking about now within populations, within species. They have tried to do so among different populations. And from my overview of the topic, I'm not saying a review, I'm saying overview, what I could find is that among populations, we don't really see this trade-off. So populations that have a high intrinsic growth rate also have a high carrying capacity. And then within populations, so among genotypes, these studies have been mostly done in bacteria and very small organisms. The evidence is mixed. So in some cases, this RNK seem to be positively correlated, in other negatively correlated, and sometimes there's no correlation, okay? So the question is actually, should RNK really trade off? And some people argue that no. So what I have presented you and the traditional way of presenting it states that these are two different environments, saturated and unsaturated, and the traits that allow you to be well adapted to one environment trade off with traits that allow you to be well adapted to the other environments. This is the original way in which it was formulated. But many people have said that this is not necessarily the case, because traits that allow for rapid growth also allow to reach higher carrying capacities in a given environment. So actually you should expect a positive correlation. And among those people is Klaus, our collaborator. And Klaus also argues in his wonderful article of 2006 in American Naturalist. I really love this article very much. I think I love it more than Klaus himself. Then Klaus himself loves the article. So basically what Klaus argues here is that RNK are very phenomenological entities or parameters. So it's difficult to actually pinpoint what is actually being selected, right? Because if you think, like, for example, in the carrying capacity, it's a number of individuals on a given environment. I mean, there cannot be direct selection for a number of individuals in an environment, right? So we should really look for the traits that underlie these different parameters and search for the correlation among these traits that underlie, these individual traits that underlie these demographic parameters. So that's what we decided to do some time ago using spider mites. I don't know if you know spider mites. They are the most wonderful study system that can exist, although some people that work with spider mites nowadays don't believe that, but others do. So why are spider mites so good to study this system, to study this question? It's because they are major crop pests and they occur in fields, and they occur on plants, and plants are very sparse, and so they colonize new plants, and when they colonize new plants, you find a very typical intrinsic growth rate curve. So there's an exponential growth, and sometimes they stay for long times on a plant, and then you find the density-dependent growth, as nicely shown by Anurag Agrawal some years ago. And moreover, they are very easily amenable to laboratory experiments. We can very easily measure individual traits and population traits. We can also do experimental evolution. I mean, they're just fantastic organisms. So that's what we did. So what did we do? We first generated isogenic lines of the spider mites, and for that we picked up an outbred population, and then we did 14 generations of sibmating, and so we have different genotypes that are fixed in these different lines. And so in these lines we measured the traits of interest. So this allows us to first see whether there is a bit of there is genetic variation, and by genetic variation I just mean broad sense heritability, so it also includes maternal effects. I'm not saying it's completely genetic, but it also allows to measure whether there are correlations among traits, because you can measure the different traits in the different lines and see whether they correlate. So this is what we did. So what did we do? First we measured what we call the population traits. So the first one is the intrinsic growth rate. So for that we took the Euler-Lotka formula that you can see there, and the two values. So this, the R is done by reiterations, and for that to have that value we need the age-specific longevity and age-specific fecundity. So for that we had one egg on a leaf. We let it develop until adulthood. That gives us the age-specific longevity. And then we let that female lay eggs during many days, and we measured each day or each 2 days how many eggs it lays. So that gives us the intrinsic growth rate. Of course, we did this with many females, right? Not just one. And also many females per different lines. And then we also measured the carrying capacity in the different lines, and for that we let we started with 10 females. We let the population grow in a sort of resource-contained environment, and then so they sort of reached a more or less stable number, and then we fit the logistic curve and extracted from that the carrying capacity. Then for the individual traits, what we did was to measure so we reasoned that the intrinsic growth rate would be correlated to the amount of resource that they ate when alone. So for that's the small a there. And for that, what we did is that we placed a mite on a leaf disc, and then we measured the damage that is produced on the plant, because spider mites produce very specific damage. It's they eat the cell content, so they take out the chlorophyll. So we have developed a method that really reads the damage that is produced and gives us a value. Developed by André himself, actually, with the help of people from the microscope department. And then we also measured the interference competition, so how much the competitive interference affected the amount of food that they eat, which is the small L there with the dashed area. And for that, we put on in the same area increasing concentrations of or densities of mites, and then we did the per capita damage of those in those different densities, and then extracted the line that characterized that decay. So first I want to show you that there is genetic variance for these traits. So the R and the K vary among lines, and so the variation among lines being higher than the variation within lines tells us that there is some genetic variance. There is also genetic variance for fecundity when alone and fecundity at the different densities. So there's also a genetic variance for competitive interference, but not for the resource use, not for feeding when alone, okay? So we did not use that trait to measure correlations, because to measure correlations you need variation in the two axes, right? So, is there a trade-off between R and K? Finally, after 11 of 12 minutes of talk. No, No, there is no genetic trade-off, there is no trade-off. The lines that actually there's a positive correlation. So lines that grow faster are also lines that reach higher carrying capacity. However, if you look at the individual traits, what you can see is that there is a negative correlation, a very clear negative correlation between fecundity when alone and the decay in fecundity caused by the presence of others. So here, there is a clear trade-off, and I was very, very happy to find this. So now the question is, why are these things unrelated? So why do we find the positive correlation in the population traits and a negative correlation in the individual traits? So we don't really know. But what we know is that there is no correlation between the individual traits that we identified and the demographic parameters that we were seeking to explain. So if you look here, that's the correlation between, I don't have my second eyes, but there is no correlation between the fecundity when alone and the intrinsic growth rate, and there is also no correlation between the carrying capacity and the fecundity and the eating in presence of others, okay? So we still don't know, but we will investigate on this further, hopefully. So in conclusion, we found genetic variation for almost all traits, except for resource use. Probably resource use is so much basal and so much correlated to fitness that we don't find genetic variance there. We found a trade-off between the underlying individual traits, but not in the population traits. We also found that these two things are not correlated with each other. So possibly there are other traits that determine or that affect the demographic traits that we are trying to measure, like, for example, the ability to wander and find other parts of the resource varies across populations. We don't know. So definitely we need more studies that will allow us to understand the underlying traits of these demographic traits so that we understand selection in saturated and unsaturated environments. And with this, I want to thank the MITE squad and you for listening. Thank you very much. Unfortunately, we have only 30 seconds for questions. Yes. Sorry. This is the MITE squad. Yes.
So why did you measure intrinsic growth rate from the life table instead of just measuring on the population growth curve?
I measured the population. From the other curve, from the curve of the K. Yeah. Yes, we could also do that, indeed. Yeah. Well, it's possible. Yeah. Thank you. So I think that was it. There are 2 seconds left. Thank you.
Good morning, everyone. Thank you for being here. And this is the last talk of the Entangled Evolution Symposium. My name is Leo. I'm from Ben Yang Leo's lab at the National Health Research Institute in Taiwan. I'm honored to be here today to present my current research on the white eye rings in rodents, their correlated evolution with diurnalism, and potential role in intra-species recognition. So in mammals, the conspicuous facial color patterns, such as the black marks, can be observed in many species, such as the dog mouse, raccoons, and panda. This feature has been explained intensively in the previous literatures to function as the concealment strategy to hide the eye position and also disrupt the facial outline. They also facilitate postmatic communication, signaling that the predator should not invest their energy to hunt the prey, and also act as the anti-glare device. In addition, many mammalian species possess the white eye rings around the eye. However, the adaptation of these traits were poorly described previously, and intuitively we can think that the white pelage tends to expose the eye position and also reflect the dasal into the eyes of the species. So what is the adaptive meaning of the white eye rings? So in this study, we hypothesized that the species with the white eye ring will be more attractive, will be sexier. White eye ring may be might provide the visual signals in the diurnal rodents to promote social communication. So to test this hypothesis, we take two approaches. The first one is the comparative phylogenetic perspective. We compare the we investigate if the presence of the white eye-ring is associated with diurnal activity patterns and with other confounding factors such as the habitat preference and social organization. And the second perspective is the behavioral perspective. We test the preference for white eye-ring in a murine model called the Nile-red Avicanthus niloticus using the three-chamber social test. So there are a couple of reasons why we focus on rodentia in our study. This order consists of more than 40% of mammalian species. They have relatively small body size and adapt to the cryptic lifestyle that tend to hide and run away from danger to avoid predation. They also occupy a wide range of ecological niches, including diurnal pattern, habitat types, and social organization. The order of the rodentia consists of 5 suborders, 3 of which are the major ones and make up of 95% of all rodent species, including like Hystricomorpha, Myomorpha, and Pseudomorpha. Interestingly, the white eye-rings can be observed in like many species among of these 3 major lineages. So in this study, we collected images from 600 rodent species from the internet source, mainly the iNaturalist. So shout out to the contributors. For each species, we then obtain their diurnal activity patterns, whether they are diurnal or nocturnal species. So noted that there are 2 other types of diurnal activities, including crepuscular and cathameral, but we omitted these patterns to simplify the data analysis. So diurnal lifestyle has been reported to lead to acquired daylight vision and improved visual acuity in rodents. Thus, our hypothesis is that the white eye-ring is an adaptive trait in diurnal rodents to promote visual communication. So we also focus in other confounding factors like the habitat preference, like whether they live in the forest, in the grassland, or desert, because we expect the white eye-ring to be found in open environments in which the white eye-ring can be easily visible. And the last confounding factor is the social organization. Is also under our consideration. The color pattern appears in the group living species to promote the conspecific, which have the animal to gather around, monitor each other while foraging in the wild, and appear in the solitary species only for mating purpose. So to test the association between the white eye-ring and each of the social ecological traits, we use the phylogenetic logistic regression for 2 binary characters. So basically this method is the logistic regression, but weighted by the phylogenetic information. So you can refer to the paper from Ives and Garland in 2010. So I won't go to much detail about the mathematics here, but just to mention, if the log ratio is positive, then there's a proof that a positive correlation association between the white eye-ring and the value x equal to 1 of the trait. So for example, if diurnal activity, I encode the nocturnal as the reference, is means x equal to 1, and diurnal x equal to 1. The nocturnal is the reference, x equals to 0, so the diurnalism is x equal to 1, so, and vice versa. So in case the log-out ratio is equal to 0, there's no association between the presence of the white eye-ring and the trait. So using this method, I found that the presence of the white eye-ring is associated with diurnalism in rodentia with a very high and positive log-out ratio of 4.6 nearly compared to nocturnalism, which is the reference in this case. This finding is consistent with the results found in 3 independent lineages. We further reconstructed the ancestral state of the white eye ring on the left-hand side, as you can see, and the diol activity pattern on the right-hand side in rodentia, and found that the most recent common ancestor of rodentia is likely to be nocturnal and absent of the white eye ring. In addition, the emergence of the white eye ring is coincident with the temporal niche shifting from nocturnal lifestyle to diurnal lifestyle. And this finding is consistent and independent among 3 major lineages. Last but not least, the earliest event of this can be dated like 35 million years ago in Shurida family of Shuromorpha. So, considering other confounding factors, we found no association between the presence of the white eye ring with any types of habitats, including forests, grassland, and deserts. And also, there's no clear evidence to support the white eye ring is associated with group living species or solitary species. So, in summary, during the temporal niche shifting from nocturnal lifestyle to diurnal activity, diurnal rodents actually adopted daylight vision and improved visual acuity. Thus, in this study, we propose that the prevalence of the white eye ring may provide visual signal to facilitate species recognition and mate choice. But given that the rodents have relatively small body size, so they require close contact to communicate with each other. So the habitat type here might not play any influence on the presence of the white eye ring. And also there's no clear evidence about the role of the role of the white eye ring in solitary and group living species seems to be similar. And as for the next perspective, which is the behavioral approach, we conducted experiments using the Nile rat Avicanthus niloticus as our rodent model. Unlike the brown rats and house mice, Nile rats are diurnal. They are social animals and possess conspicuous white eye ring in both genders. Furthermore, my research group previously demonstrated that the Nile rats acquired daylight vision and improved visual acuity, but with the degeneration of olfactory system, so-called the vision and olfaction trade-off. Thus, our hypothesis is that if the olfactory system is degenerated, vision becomes more prevalent and become more important. So the white eye ring here can step in to serve an additional signal for visual interspecies communication in the Nile rats. So to test this hypothesis, we adopted 3-chamber sociability test. The apparatus is divided into 3 parts: the central chamber, chamber A, and chamber B. So the behavioral test consists of 2 phases. The first is the habituation phase and testing phase. Each of the phase consists of 10 minutes of observation. So during the 10 minutes habituation phase, we introduce the Nile rat subject into the central part of the chambers and allow them to freely access to the whole chamber. And prior to the 10-minute testing phase, we install the images of the Nile rat with normal white eye ring in the chamber A and with the masked white eye ring in the chamber B. So using Photoshop, you can see that it's clearly the white eye ring is disappear. So we try not to use the actual animal to eliminate all of the olfaction cue that can be introduced in our experiment. So the time spent in each chamber, chamber A and chamber B, during both habituation phase and testing phase was measured. So we calculate an index called the strength of preference, or the SOP, for the white eye ring to evaluate whether the Nile rats are attracted to the white eye rings or not. The index was calculated based on the log ratio of time span in chamber A and chamber B during the testing phase, and corrected by the side bias introduced in the habituation phase. So if the SOP is higher than 0, there's more preference in chamber A, and so that the Nile rats are attracted to the white eye ring and vice versa. In case the SOP is not deviated from 0, there's no substantial preference for the white eye ring in the Nile rats. So here's the result. Oh, sorry. So when being introduced with male or female Nile rat images, both male and female animal subjects showed significantly stronger preference for the white eye ring. So you can see as the SOP is higher than 0 in all of the treatment and all of the in both sex. So we can conclude that both male and female are attractive to the white eye ring, no matter which sex of the image were introduced. Moreover, there was no significant difference in the median of the SOP between male and female samples in both treatment. So we conclude that the white eye ring may play a role in conspecific or mate choice, but not sexual selection. And given that this white eye ring can be observed in both sexes of the Nile rat. So in conclusion, using both methods, comparative and behavior methods, we found that we provide evidence to support the correlated evolution of the white eye ring and diurnalism, but not other ecological traits like habitat or the social status. The presence of the white eye ring emerged simultaneously with the transition of the nocturnal lifestyle to the diurnal temporal niche during rodent evolution. And we also proved that the Nile rats without the white eye ring are less attractive, and male and female Nile rats show similar interest to the white eye ring. So in conclusion, the white eye ring may be linked to enhance visual acuity and could enhance the species recognition and mate choice in diurnal rodents. So I would like to acknowledge my lab mate, my PI, Dr. Ben Yang Liao, and our collaborators to support this project. And also our funding agency, the Taiwan National Science and Technology Council. So this is my reference. Thank you for your listening. And if you are interested in the Nile rat, please feel free to contact me. Thank you very much. So do you have any question?
I wanted to ask if you have done the choice experiments with animals that have an exaggerated Photoshopped white eye ring as well, because I could also imagine, kind of as an alternative hypothesis for what you see, that they like animals that look normal, more than that it's something to do with the exact eye ring.
Thank you very much. That's a very interesting question. Actually, I'm also planning to use other species like the brown rats on the house mice and paint them with the white eye ring to see what is the behavior of the Nile rats. So let's see what happens later. Thank you very much for your suggestions. Any other question? Okay, thank you very much. So I think this is the end of the symposium.